Hello friends, welcome to BMH Learning. This video is about the Miles and Misra method. The Miles and Misra method is a microbiological technique used to determine the number of colony forming units, or you can say CFUs, in a bacterial suspension. Unlike other cell count techniques, Miles and Misra method is actually a surface viable count method. That is, it determines the number of alive or viable colony forming units. The method is named so because it was first described by Miles, Misra and Irwin in 1938. Materials to be required: a calibrated dropping pipette which can deliver drops of 20 microliter, petri dishes, nutrient agar or other suitable medium, phosphate buffer saline or any other appropriate diluent can also be used, and most important, a good bacterial suspension. Procedure. So, first thing we require is bacterial culture or inoculum or suspension, we can say. The inoculum is in liquid media. We need to make 10-fold dilutions of bacterial suspension. For that, we are using phosphate buffer saline as diluent. To prepare 10-fold dilutions, we take 1 ml of bacterial suspension and 9 ml of PBS. From this tube, we take 1 ml and transfer to next tube, and the process is repeated for each tube. This way, we get 1 in 10, 1 in 100, 1 in 1000, 1 in 10,000, and this goes on. Let's suppose we are only making 4 dilutions. Now, petri plates are taken. Molten nutrient media is poured into these plates. The surface of the plates should be dry enough to absorb the bacterial dilution within 15 to 20 minutes. Now, this plate is divided into 4 sections: A, B, C, and D. The number of divisions can be increased depending upon the number of dilutions made. We need 3 plates for each dilution series. For statistical reasons, an average of at least 3 counts are required. For simplicity, I am only showing here 1 petri plate. The sections are labeled with the dilutions. In each division or sector, from a height of 2.5 cm, 20 microliter of the appropriate dilution is dropped onto the surface of the agar, and the drop is allowed to spread naturally. From this much height, the drop naturally spreads over an area of 1.5 to 2.0 cm. It is very important to not touch the surface of the agar with the pipette. The plates are then left to dry and are then allowed to incubate at 37 degrees centigrade for 18 to 24 hours. After incubation, we observe each sector for bacterial growth. Sectors with high concentrations will give a confluent growth over the area of the drop, or we can say you would notice a large number of small or merged colonies. We will count the colonies in that sector where we will observe highest number of full-size discrete colonies. We use following equation for the calculation of the CFUs per ml from the original aliquot or sample. CFU per ml is equal to average number of colonies for a specific dilution into 50 into dilution factor. This was all. Thanks for watching.